DNA was initially considered too simple to serve as a mechanism of heredity, but in fact its chemical properties make it an ideal medium to store genetic information. DNA is composed of nucleic acid, one of the four major classes of biomolecules along with carbohydrates, proteins, and lipids. Nucleic acids consist of deoxyribose sugar, a phosphate group, and one of four nitrogenous bases, adenine, cytosine, guanine, and thymine. RNA is similar to DNA, but uses ribose as the sugar and basil instead of thymine.
RNA is single-stranded and does not form a double helix, but RNA strands can fold back on themselves and form complex three-dimensional structures. Therefore, RNA is in some ways a more interesting molecule than DNA. It is used by some various to store genetic information and can also perform some of the catalytic function of proteins. RNA probably serves as the genetic material in the earliest organisms, but the double-stranded nature of DNA makes it superior as errors can be rapidly detected and repaired.
The ability of DNA to encode information is a direct result of strict pairwise interactions between guanine and cytosine, which has 3 hydrogen bonds, and between adenine and thymine, which has only 2 hydrogen bonds. This pairing is required for proper spacing of the strands. These double versus triple hydrogen bonds provide specificity but also differ in the amount of energy required to break the pair bonds. As a result, regions rich in adenine and thymine are more flexible and separate more easily. They are often found within promoters and regulatory regions and at origins or replication.
Unlike the weak hydrogen bonds holding the strand together, the nucleotides forming the DNA backbone are covalently bound by a strong phosphodiester bond between phosphate and sugar groups on adjacent nucleotides. This strong backbone is essential in order to prevent the DNA strand from fragmenting. Chromosome 1 alone contains 248 million bases, forming a molecule 8.5 centimeters long. DNA resembles a spiral staircase, but it would be an awkward stairway to climb, as the helix alternates between major and minor groups. What information does DNA actually encode? Very little, as it turns out. Only about 1% of the genome code for the approximately 20,000 protein coding genes.
This was a surprise, as it was assumed that the genome should contain about 100,000 genes to account for the much larger number of observed proteins. However, the discovery of introns and alternative splicing made this one gene, one protein model obsolete. Although splicing is a complex process, and nearly 1% of all genes play a role in splicing. Then, what does the other 99% of the genome do?
The rest of the genome consists largely of repetitive sequences. Some of these regions play important structural roles, such as the centromeres and the telomeres, but much of it consists of pseudogenes, artifacts from ancient transcriptional activity, and other DNA often described as junk. However, this apparent junk DNA is often found to perform important regulatory roles and may be transcribed as long non-coding RNAs or microRNAs. Regardless, even the DNA that ultimately serves no purpose at all is still an integral component of our chaotic genome, and we would not be who we are without it.